Hi everyone, this is Volodymyr Krout, and today we will talk about microbial inoculants for legumes. Inoculants is a group of products that take pride of place in the technology of growing legumes. In fact, the term inoculation has a broader meaning. Inoculation is the introduction of microbial mass into any environment. You can inoculate soybean or sunflower seeds, land or plants. From a technical perspective, even taking probiotics with living organisms is also inoculation. But today, we'll talk specifically about inoculation of legumes with symbiotic nitrogen fixers. The role of nitrogen in the formation of all protein compounds is known to everyone. But how exactly this nitrogen becomes available to microbes and plants is an intriguing question. Scientists say that about half of the nitrogen in each person's body comes from mineral fertilizers. The other half is obtained naturally due to the work of nitrogen-fixing microorganisms. All the nitrogen that microorganisms get from the atmosphere is fixed with the help of several enzymes, the key of which is Nitrogenase. This enzyme allows to break the super-strong bond of two nitrogen atoms that form nitrogen and two molecules and transform them into two ammonia NH3 molecules that all plants adore. But there is a gray area. The work of nitrogenase is impossible in natural conditions, so free-living nitrogen fixers have to spend their energy on inactivating oxygen molecules from the air. Instead, symbiotic nitrogen fixers that form nodules with legumes rely on the plant when it comes to the protection against oxygen. The roots of legumes form a specific protein, legumoglobin or legoglobin. By the way, it is similar to human hemoglobin. This protein binds excess oxygen in the roots of the plant and allows symbiotic nitrogen fixators to work. I might add that the rich red color of active nodules is associated with the presence of this legoglobin. It is due to this protection that symbiotic nitrogen fixers demonstrate much higher nitrogen-fixing efficiency than their free-living colleagues. So, what type of microorganisms are able to carry out the nitrogen fixation process in the soil? Among the free-living bacteria, the most famous but not the only ones are representatives of the genus Azotobacter, Bacillus, Klebsiella. They are able to leave behind about 20 kilos of nitrogen per hectare per year. Besides them, there are also associative nitrogen fixers, something average between symbiotic and free-living microorganisms. They do not form specific organs with the plant, but are able to fix atmospheric nitrogen only in the rhizosphere, where the plant guarantees them the presence of sugars, growth factors and low oxygen pressure. Such associative bacteria include representatives of the genus Azospirillum. But we are still interested in symbiotic nitrogen fixers. Everyone knows that each legume species has its own specific type or even a variant of the symbiotic bacterium species. For example, soy forms symbiosis with Bradyrhizobrium japonicum, peas with Rhizobrium leguminosarum biovar VC, beans with Rhizobrium leguminosarum biovar phaseale, lucerne forms symbiosis with Cynorhizobrium meliloti and lotus with Mesorhizobrium loti. The secret of such pickiness in choosing a neighbor is hidden in the complexity of the process of forming a nodule. And now I'll tell you about it. Usually, the plant releases flavonoids, specific biologically active substances, into the soil. In response to them, nitrogen fixers move in the direction of the root of the plant, namely to its root hairs. The bacterium attaches to them with the help of other specific substances that are released by both themselves and the plant. Now, the bacterium manifests itself to the plant with the help of special modules, nod factors. These nod factors cause the root hairs to twist, and bacteria at this time penetrate into it. There they form an infectious thread. When the infectious thread reaches the root, the plant begins to form its own nodule, surrounding bacterial cells with its tissues. Bacteria, on the other hand, lose their own cell wall and come into direct contact with plant cells, gaining direct access to the sugars, oxygen and nitrogen bound by legoglobin. Now, the bacterial enzyme systems are ready to supply the plant with ammonium nitrogen. Therefore, in order for a nodule to form, a long chain of molecular interactions between the plant and the bacterium must work out. Among other things, nitrogen fixation requires significant amounts of energy. It is estimated that you need to use about 25 kilos of dry matter in order to fix 1 kilo of atmospheric nitrogen. And if we take into account that symbiotic nitrogen fixators can provide up to 200 kilos of nitrogen per hectare during one growing season, it becomes clear why symbiosis with an active synthesizing plant makes them much more effective than free-living microorganisms. That's all for today. I hope the information was interesting and helpful. Subscribe to our channel, smash the thumbs up, and see you in the next videos.